காடுடைய செவியன் விடையேறியோடுடன் மதிசூடி காடுடைய சுடலை பொடி பூசியன் உள்ளங்கவர் கல்வன் ஏதுடைய மலரானுனை நான் பணிந்த அருள் செய்த பீடுடைய பிரமாபுரமேறிய பெம்மான் இவனன்றே.
Dream is not something that you see while sleeping. It's something that does not let you sleep. Good afternoon, one and all present here. Seminars are organized and conducted to gain insight and information that would remain effective in their life, exercises, and work. On behalf of the Department of Mathematics, I extend a warm welcome to the management of CMS College of Science and Commerce, who motivate us in all the endeavors of the college activities. Today, we have the resource person, Dr. K.PARVATHAM ma'am, Professor, Department of Mathematics, and also IQAC Coordinator of Sri Sai Ram Engineering College, Chennai. I consider it as not only my privilege, but the honor of our department to host her as a resource person. On the behalf of everyone present here, I cordially welcome you, ma'am. Welcome you, ma'am. I would like to welcome Dr. T. Vinod Kumar, Director, School of Biological Sciences and DBT Star College Coordinator, who has contributed a lot for the DBT Star College scheme of our college. Welcome you, sir. I extend a warm welcome to Dr. Mridula Ravindran, who motivated us in all the aspects of the department work and college work. Welcome you, ma'am. I am extremely honored to have the chance of welcome the faculty members of the department who stood behind the success to make all the programs a successful one. Now, I extend a warm welcome to all the mathematics department students and bioscience department students who have joined for the webinar. I hope and request your willing cooperation throughout this program. Once again, I take this opportunity to welcome you all to this program. Thank you.
Thank you, ma'am. It was indeed a warm welcome to you. Now I welcome Ms. Shalini R, Assistant Professor in Mathematics, to introduce our beloved chief guest of today's session.
Good afternoon, everyone. It is my privilege to introduce the resource person of today's program, Dr. Parvatham, Professor, Department of Mathematics, IQSC Coordinator, Sri Ram Engineering College, Chennai. She has completed PG from Bharatiyar University in the year 1988 and MPhil from Annamalai University in the year 1990. Her specialization in MPhil, generalized inverses of matrices. She completed PhD from the University of Madras in the interdisciplinary domain Mathematics Zoology. She also completed the certificate course in bioinformatics from MIT campus of Anna University. She has published 10 papers in national, international journals and conferences. She has teaching experience of 28 years in engineering, arts and science colleges. Completed nearly 20 online certificate courses, obtained SWAYAM NPTEL domain certification in faculty domain advanced. She has conducted webinars on mathematical biology and NEP 2020. She has invited as a keynote speaker to VIRC in the year 2018, organized by the Faculty of Science, University of Jaffna, Sri Lanka, and co-organized by Ministry of Science, technology, research, skill development, and occasional training, and Canadian Heritage, Sri Lanka. We are very fortunate to have you, ma'am, among us today as a resource person. Thank you, ma'am.
Thank you so much
Thank you.
for your nice introduction. And say I'm from Sairam Engineering College and not Sriram. It's always it happens. Okay, hope I can start or is Praveen?
Ma'am, one
Can
second.
start or you have Imo?
Yes,
Next,
ma'am.
Okay.
next I welcome Dr. Pavadam, Professor, Department of Mathematics, Coordinator IQAC, Sri Sai Ram Engineering College, Chennai for the technical session.
Yeah, thank you. Thank you so much. So I think it's good afternoon to one and all here. Thank you so
Good
much for
afternoon,
your
ma'am.
invite. And I take this as a great opportunity to share my little knowledge, whatever I have with the students of both mathematics and biotechnology. I think you have biotechnology, biochemistry, all the students are here, is it? Can you know what are the branches you have in this webinar? Am I audible?
Yes, ma'am.
Yeah, you have biochemistry, biotechnology, all the students here? It's a
Yes,
combination
ma'am.
of,
Yeah, combination of biosciences, ma'am.
okay, okay.
Nutrition is also there, ma'am.
Okay,
Nutrition
okay,
so
okay, okay, thank you, okay, okay. See, it's a post, not a post lunch session. It's a pre lunch session. It's both difficult for the speaker as well as the audience. Let me try to make it interesting. So here I'm going to say about mathematical modeling and explain some three mathematical models. Can I share my screen? Yeah, is my PPT visible? Is it visible?
yes, ma'am. Yes, ma'am.
Okay. So the topic I've chosen is biology in the language of mathematics. You can, if you search in the website, you can even see a paper named Mathematics is the Next Microscope of Biology. So better some starting like this. Mathematics is the next microscope of biology. So that much mathematics is important for biology, and both the biology students should learn mathematics and mathematics students should know biology. Only the collaboration of mathematics and biosciences students, you can come up with very good projects and you can come up with very good new findings also. So the outline of my today's talk is, so I'm going to say what are mathematical models and why we are going for mathematical models. And you're going to see three different models. One is the model of environmental biology and ecosystem. And the next one is the models of epidemic diseases. And the last one is the models of metabolic networks. So I'm not going to do in depth about solving this mathematical equations and all. Just I'm going to explain how you form this mathematical models. Let me make it so simple, not going very much deep into mathematics, because I'm from mathematics background, but I have an interest in biology. So when this interest came as in my MSc, I read a paper that is mathematical simulation of blood flow near branched arteries. You can, if you Google this, you can find the full paper of this also. So this paper created an interest in biology. It's actually with fluid dynamics. The work will be with fluid dynamics. So this paper only created the interest for my biology. That is to link, to bridge between mathematics and biology. And luckily, I got an opportunity to do my research also in that topic. Okay, now, so we are going to see this. Now, what are mathematical models and why are we going for mathematical models? See, mathematical models, they are nothing but the representation of some real world scenario in terms of mathematical equations. So you put some natural phenomenon in terms of mathematical equations. See, this mathematical model is not new to you people. It have been introduced in the schooling itself, in your high school itself. So, for example, you take a problem like this. That is, Suresh got some 4 apples and 3 oranges for 55 rupees. And say some Ramesh got some 6 apples and 2 oranges for 70 rupees from the same shop. Find the cost of the orange and apple. So you would have done problems like this. So they are nothing but some real scenario put in the form of a mathematical equation. So that's a mathematical model. So mathematical models are not so complex. It's only an easy thing. But many people think that when you talk about mathematical model, it is something very complex, and people find it uncomfortable with models. So mathematical models, they are nothing but representation of a real world scenario in terms of mathematical equations. Now, why do we go for this mathematical models? So what is the use of this mathematical model? Why do you want to go for this mathematical model? So it is just to make a prediction about the possible solution. So you can extrapolate the things using this mathematical modeling. So today it is like this. What will happen in the future? So you can extrapolate things using the mathematical model. So your mathematical models in climatic conditions, that is global warming and all, they use mathematical model very much. So to predict the future, you can use the mathematical modeling. And mostly mathematical models are open-ended problems. So what do you mean by an open-ended problem? What is an open-ended problem? Open-ended problem means there exists more than one solution. You may have more than one solution for that. So mostly the mathematical models in natural scenario, they'll have more than one solution. That's why people find it very uncomfortable. So whenever you go for this mathematical model, the assumption what you make in the modeling is very, very important. Your assumptions play a vital role in these models. So whatever solution you get for this, that should satisfy the assumptions. So that's a very important criteria while modeling a real world phenomenon. Okay, so this is about your mathematical model. So mathematical model mainly, they make a prediction about the possible solutions. So you can predict the future using a mathematical model. Now, let me go to the first model. So can you guess what is this? So what's the phenomenon happening here? Can any of you guess? Yes, you can unmute and say the answer. So I'll not be able to see the chat window because I'm presenting here. You can unmute and answer. So I want an interactive session. I prepared a quiz also at the end. Yes, can you guess what is the phenomenon happening here? Any of you?
Students, please.
Pardon?
Students, please respond.
Is what's happening here? Yes. See, one living being is eating another one. So what comes into your mind when you think of this? Is biology students are here? What comes into your mind when you think of this? So this is the first model we are going to see. That is your predator-prey model. Hope you have heard this. The predator-prey model. That is, you know what's a predator and you know what is a prey. So how this model came into existence, your predator-prey model, how this model came into existence and how this model was constructed and what is the story behind this model. So this model is named as the Lotka-Volterra model. See, the 3 persons were involved in constructing this model. So first one is this Vito Volterra. He's a famous Italian mathematician, and his son-in-law was D. Ancona. He was a biologist, and Lotka is an American mathematical biologist. So actually what happened is, Humberto Ancona, he was studying about the various species of fish in the Adriatic Sea. So he was in the Adriatic Sea, he was studying about the various species of fish. So what he did was, he made a statistical study of the various types of fishes sold on the fish market. He took 3 ports. Okay, you can see the ports in here also in Triplicane, and somewhere else we have the ports. So fish sales will be more there, and fish markets will be found near the port. So he studied the various species of fishes that were sold on 3 ports. He took that into account. This person, Humberto de Ancona. And he also studied about the percentage of predator species. So you know what's a predator and what's a prey. So predator will eat the prey. That is, in the fish itself, you have some fish will eat the other fish. So you have some species. So for example, if you take the shark will eat the other fishes also. So shark is called as a predator. Okay, a shark is a predator. So he studied about the predator species also. So he studied the predator species also sold in the market. So what happened was, this was the details he got. That is, in the year 1914, 12% of predators. This he took the port name is Fiume fish catch. And 1951, 15 it is 21, like that the data goes on. He has taken from 1914 to 1923. These years you have noted down, and this is the thing he got in his statistical study. Now, what he observed in this is, so see, World War One. When was a World War One? It was in the period from 1914 to 1918. So he observed that the highest percentage of predators were during World War One. So during the World War One, that is from 1914 to 1918, there was the highest percentage of predators, and also just after the World War. So this was he observed in his statistical study. He observed that the highest percentage of predators recorded during World War One or just after the World War. That was in the year 1919. So I think World War started in the middle of 1914 and ended in the I think in the month of November, 1918 November, it got ended. So during this and immediately after you end of the World War. So this was he found out. And during this period, during this World War period, the catching of fish was limited. Fishing was limited during this period. So what he concluded is there was a predator-prey balance during World War. So he concluded that there's a balance between the predators and prey during the World War, and an intense fishing, that is before and after the World War. So when there is more of fishing, this natural balance was disturbed. That was his conclusion. So is it clear? So his conclusion from his study was during the World War, there was not much of fishing. So people were not allowed to go to the fishing. So during that time, the predator-prey balance was maintained. And when the fishing started, that was before the World War and after the World War, when again fishing started, this balance was disturbed. So this he found out. So what he did is, the son-in-law, he went and asked his father-in-law whether he can represent this natural phenomenon in the form of a mathematical model to explain what is going on, why this balance is created and why this balance was disturbed. See, he wanted to put it in the form of some mathematical equations. So he went and talked to his father-in-law, because his son-in-law is a biologist and father-in-law is a mathematician. So he went and asked him to just know about this model and to explain this mathematically, what is happening, and can he predict about this in the future. So he went and asked him. So in the meantime, this person, this Alfred J. Lotka, he is an American mathematical biologist. He was also working on the same type of problem, but what he was studying is that a plant population and an herbivorous animal dependent on that plant for food. Okay, he was not studying about the fishes, and he was studying about a plant population and then an herbivorous animal that is dependent on that plant for food. He was studying on this. So now these three persons, they joined together and they formed this Lotka-Volta model. So already I told you that the assumptions are very, very important in a model. So when you form the equations, your mathematical equations, the assumptions what you make are very, very important. Even in that example, what I said, I told you they are buying the oranges and apples in the same shop. If they are buying in some other shop, the rate may vary. So it's not possible to find the exact value of the orange and apple. So that the assumption what we made is they are buying it in the same shop. The same way here, what are the assumptions they make here? Your assumptions play a vital role in finding the solutions of a mathematical model. So the assumptions what they made is The rate at which predators eats the prey is jointly proportional to the size of the two population. Hope it's clear. The rate at which predator eats the prey. So, say if there are more prey, a predator will also eat that. Okay, it is proportional to the size of the two population. If the preys are very less, the predator may not get that much of food. Okay, predator will eat the prey. So, for example, you can take fox as a predator and rabbit as a prey. Okay, and the other thing is a fixed proportion of encounters leads to the death of the prey. So, when a predator eat the prey will die. So the prey population will reduce. So these are the two assumptions you make here. Now he assumed that X is the prey population, X of T. Okay, so X of T is nothing but T is for the time. It's the prey population at time T in a particular time interval. Okay, so say from some in 1 day period, in 24 hours, the prey population is taken to be say X of T. T is nothing but your time. Okay, X of T is the prey population at time T. Now, your differentiation, that is dx by dt. So biology students don't get afraid of this dx by dt, this calculus, it's nothing but rate of change of one variable with respect to the other. So here dx by dt means it is nothing but the decrease in prey population, okay, or the increase in prey population in a particular time period. So when you will have an increase in the prey population, when there is no predator, there will be an increase in the prey population. Okay, and then whenever there is a predator, there will be a decrease in the prey population. When some predator comes and eat the prey, you will have a decrease. So this is dx by dt is nothing but the rate of change of prey with respect to time. Okay, so this is proportional. So that proportionally constant is your A. Okay, X of T is the prey population and this proportionality constant is your A. So there will be so when you will have a negative component means in the prey rate due to the predators, whenever there is an encounter, your prey, there will be a negative component in the prey population. Clear? So that's why you have this as minus bx. So in the starting, that is a time, initial time, say when time t equal to 0, I may have this as dx by dt is equal to some ax. So after some, the time passes away, say there are 5 rats in the beginning, and after say some 10 minutes, 2 rats has been eaten by the fox. So in that case, what happens? The number of prey will reduce. So I'll have minus b. There will be a negative coefficient here. So minus b and x stands for the prey population, and because of this assumption, it is jointly proportional, and your y is nothing but the predator population at time t. Okay, your y is the predator population at time t. So I have this equation dx by dt is equal to ax minus bxy. So this is very simple. When you put it in mathematical, you have this equation dx by dt is equal to ax minus bxy, whereas biologically this means that, so x is the population of the prey at time t. Okay, when time goes on, when there is an increase in time, when time changes, this population, so at initial time you have this population, when times go on, when there is when the predator comes and catches the prey, there will be a decrease in the prey. So you have this minus bxy, that's all. So this is your first equation. This is for my prey, dx by dt. Now I'm defining this y of t to be the predator population at time t. So what happens? My dy by dt. So here, if there is no food, your predator will die. Okay, so predator depends only on prey for its food. It don't have any other source. That's an assumption. So the predator depends only on prey for its food. So if there is no food, your predator will die. Okay, so the death rate is proportional to the size of the predator population. If there are more predators, you need more prey. If the predator population is less, then in that case, the prey will be saved. So you have dy by dt is equal to minus cx. Okay, but because why it is minus cx, x is the rate of the prey population. So y is the rate of the predator population here. So when I say dy by dt, that is the predator will encounter the prey. So the prey population will decrease. So I have minus cx here. But here there is food, and what is bad for rabbit is good for fox, is it not? So a fox can eat the rabbit whenever it is available. So you have, again, using the proportionality of both the predator and the prey population, you have dy by dt is equal to minus cx plus p xy. Is it clear? So these are the two equations you've got. So here, what does it mean here? This equation is, that is, this is for the predator. So whenever there is a predator encounters a prey at any particular time, there will be a decrease in the prey population, and then for the predator, it is if the predator gets a food there, so you have plus p into xy, where this a, b, c, p, they're all nothing but some constants. So this is what we call it as the Lotka-Volter model. So dx by dt is equal to ax minus bxy, and dy by dt is equal to minus cy plus pxy. So this is the mathematical phenomenon of the predator and prey. Okay, this is your Lotka-Volter model. So these are nonlinear equations actually. I'm not going to solve this equations. So solving this equation is nothing but getting the value of your x and y. So getting the value of your x and y means it is, you know, what is the exact rate of the your prey population, and your y is the rate of the, that is the predator population at some time t. The solving part I'm not going to do it now. It's just how you form this model. Okay, just let me stop share it for a minute. Yes, do you have any questions? Do you have any questions here? Shall I continue? Yes, students, did you follow anything? Okay. Okay, so now what are the applications of this? Why do you go for this model? So this Lotka-Volter model, it is used to study the dynamics of natural populations of predators and prey. See, sometimes some of the species are going to get extinct. That is, they are not at all present. That is, all the species have died. So in that cases, you can use the this Lotka-Volter model to study the dynamics of this population. Okay, and this is used to predict the outcomes of the, sorry. This is used to predict the outcomes of the competition based on the impact of one species on another. Okay, so when there are when there is more of predator species, the prey population will decrease. So you can predict the outcome of the competition based on the impact of one species on another. And recently they have used this in the study of cloud behavior and marine bacteriophage dynamics, and many more applications you can find for this model. Okay, so this is your Lotka-Volter model, and now this is the basic model what I've explained. So now they have made some changes in this model according to their study. Okay, you can change the parameters according to your study, and you can find the solution. Whatever problem you have, accordingly you can form the model, and you can study about that. Now the next model, what you're going to see is, I told you it's about the epidemic, it's about a disease model. So here there are three persons. So this is a susceptible person, this is an infected one, and this is a recovered one. So you have here, so my S, this is mathematical model of infectious diseases. Okay, so my S is the susceptible model, your I is the infected, that is number of, that is the fraction of people they are susceptible. What do you mean by susceptible? Even during this corona period, you would have heard this word susceptible often. So susceptible is nothing but they may get exposed to the infection. Currently, they are not infected, but they may get infection. Okay. And next one is the infected people. Once they get infected, they have to get recover. So this is your recovered. So just 2 minutes, sir. Maybe now webinar better. Okay, so you have this three types of people. You call this as the SAR model. Okay, this is your SAR model. Okay, now this is a graph related to this SAR model. So you have this SIR. This blue stands for your S. That is, here it is the number of people. So your graph here is like this. So it decreases and go on. That is, the susceptible will get infected. Okay, so it comes like this. And the next one is you're infected. Those are not infected, it will go to the peak, get infected, and they get the immunity, and then they come back. This is your I. And my R is nothing but the people, recovering people. So my recovering will always be an upward curve like this. So these are the three curves you get for this SIR model. Now, how do you get this curve? What is the phenomenon between beyond these three curves? How do you get this curves? What are the equations responsible for these three curves? So that you are going to see in the SIR model. Okay, so the model itself is named as SIR model. If you Google as SIR model, you can find a lot of materials using this. This model was first found by this Kermack Megderick model. So these are the persons who were responsible to construct this model. So here again, you have the same thing what I have done for your the previous one. So my S of t is susceptible at time t. And this is infected at time t. The people who are getting infected at time t. And this is the people who are recovered at time t. So the total, when you add all this, that should be n, it is a total population. Okay, either they should be recovered or they should be infected or they should be susceptible. And here again, your assumptions. These assumptions are very important. The assumptions what you make are very, very important. So you say that there are no births during that period of time, and there is no immigration. That is, people don't immigrate from one place to another place. Okay, these are all about infectious diseases. So just infectious diseases, you think of your corona. So when they immigrate from one place to another place, there will be a new infection there. And then another thing what they assume here is your recovery gives total immunity. So recovery gives total immunity means once you get infected and you got recovered, again, there is no chance of getting that infection. So that is the assumption you have here. Recovery gives total immunity. And then there is a fixed infection rate. And again, you have a fixed recovery rate, and the people are all well mixed. So these are the assumptions you make in this model. So for this graph, so I've seen these three graphs. These are the equations, three equations responsible for that graph. Okay, again your S, I, N are here. That is susceptible, infected, and this is your recovery rate. Again, so now how do you form these equations? So now when you take the initial time at t equal to 0, let me take when t is 0, it is my S of 0 is S naught, I of 0 can be taken as I naught, and in that case my recovery, there will be no one to recover, so that value will be equal to 0. That will be my initial condition. Okay, now how do I get this first equation? Again, it is the rate of change of the susceptible people with respect to time. Okay, it is the rate of change of susceptible people with respect to time. So, for example, if there are 10 people in susceptible, that is a compartment, you can take it as 3 compartments. One is susceptible and the other one is infected and the third one is recovered. Okay, so if there are say some 10 people in the susceptible compartment, so out of the 10, some people should have got infected. So only when susceptible people get infected, they'll come to the infected category. So in that case, what happened? The number will reduce. The number of susceptible people will reduce. So I have this minus beta. Because there is a reduction of S, I have this minus beta is associated with this I, and this people are getting infected, so I have this I. So my first equation is ds by dt is equal to minus beta into Si, where this beta is nothing but the number of contacts infected per day is my beta. So my beta is nothing but number of contacts infected per day. This is my beta. This equation is for my susceptible. Now, what happens when people get infected? So what is the infected rate? The rate of change of infected people with respect to some time. So here I have positive BSI because people are getting infected. So at this point, people are getting infected, and infected people will recover. Is it not? Once they get recover, infected people will recover. They will have some recovery period. Say within a week they may get recover, or within some 15 days or something like that they may get recover. So this is, I have the infected people will reduce, and that will go to the recovery. Okay, your infected people, number of people infected will get reduced. Okay, so will get reduced means what? They will recover. So I have this minus gamma into I. Okay, so this gamma is the number of recoveries. Okay, number of recoveries per person per day is your gamma, whereas this beta is the number of contacts per infected person per day. This is a beta and this is a recovery. So from infected, when you get this recovery, there is a decrease in the number of infected persons. And you also remember this assumptions. So once there is a recovery, again they are not getting infected. Recovery gives total immunity. That is the assumption we made. So you got minus gamma into I. And this is dr by dt is, it's only the positive value here because the number of recovered cases will increase. So these are the three equations. So when you, again, these are all also nonlinear equations. So when you solve these equations, I get the value of my S, I and T. So that will give you the graph. So using those values, you have the graph here. See, if any of you are interested, if there are PG students here, both even biology students and also mathematics students, if PG students are here and if you have any projects, you can do this type of modeling projects. So it's a very, very simple model. In your PG level, you can do this. You can do some clinical study, get the values, and then you can do for this. So this is the SIR model. Now this is the basic model I'm explaining. Now this model has been become SEIR is also available now. So SEIR, E stands for the exposure. Okay, so that model is also available, and there are many parameters have been changed, and these assumptions they can change as per their problem, and this model has also got many applications, and mainly now in this corona time, this model has been used to predict the spread of coronavirus. So these are the, it helps to predict the spread of the disease, and this is used for your COVID-19. That is the prediction for COVID-19. So you can see many papers using this SAR model predicting this COVID-19. So this is the main application of your SAR model. So SAR model, it is mainly used to control the spread of the disease. So this is an infectious disease model. So this is my SAR model. That's the second model you have seen. So both these models deal with differential equations, and both are nonlinear differential equations. So I'm not going into the solution of these equations. So when you solve these equations, just like in the previous case, also you will be getting the value of this. Here you'll be getting the value of S, I and T. That is number of susceptible people, number of infected, and then here it is number of recovered people after some time t. So currently what you have, and you can predict, that is you can extrapolate the thing after some particular time, say after some 6 months, after 1 year, whatever it is, what will be the range. You can have the approximate prediction here. Okay, that's how this model is used. Now these two models are based on a differential equations. And the third model, what you're going to see is, if biochemistry students are here, they can identify what is this. What is this actually? Yes, any of you? What is this? See, this is essential for each and every human being. See, glycolysis, which is responsible for your energy production. If there is no ATP production, then you cannot live. This is responsible for your energy production. So this, you call it as a metabolic pathway. So starting from glucose, this happens in everyone's body. So this glucose should be converted to energy. So this is one pathway. This is glycolysis. And here I can see, this is glycolysis I have, then TCA cycle is there. then phosphate pathway is there. So some 3 or 4 pathways are mixed. And here you got many more pathways. When you have many more pathways like this, so the combination of all these pathways, you call it as a metabolic network. So even you have many more networks also. Social network, this www network. So many networks you have. So okay, then signaling pathways network. Many network are available. We are going to say about the metabolic network. All the networks can be modeled mathematically. Even your social network, everything can be modeled using mathematically. So this we are going to see for a metabolic network. Now this is a biochemical network. This has got 2 types of models. One is the qualitative model and the other one is the quantitative model. You know what's the difference between qualitative and quantitative? So this cannot be measured, whereas this can be measured. Your quantitative models can be measured here. These qualitative models are mostly based on your graph theory. Okay, so qualitative models are based on graph theory, whereas when you go for this quantitative models, again it is divided into 2 types. One is the kinetic models. So you would have studied this Michael-Menden's equation and the other things. So your kinetic models are based on that. You can calculate those constants. So again, the mathematical tool used in that is ordinary differential equation. So ordinary differential equation is the mathematical tool used in this kinetic model. And again, when you go for the stoichiometric models, the tool you use here is linear algebra. That's a matrix algebra you'll be using here in this. So this why we prefer the stoichiometric models than the kinetic models. So why do you prefer this stoichiometric model is this stoichiometric model, it does not need any experimental data. Okay, just you can form the model just from this biochemical equations, this reactions. From these reactions, you can form the model. Whereas when you go for kinetic models, you need the value of the Michael-Menden's constant. So the experimental data are required for this kinetic model. So mostly mathematicians, they prefer this stoichiometric model instead of this kinetic model. Okay. Now again, I'm not going to explain this. I thought I'll do this kinetic model also, because again, it's an OD based model. Let me go to the stoichiometric model. Now here, this is a biochemical reaction. Okay, I have an active cell. So what's the biochemical reaction? This takes place in everyone's body. So molecule A is converted to molecule B by the catalyst, that is an enzyme. That is, a substrate is converted into the product. Okay, a substrate is converted into the product by the action of this enzyme. So this is from a biologist view. Okay, the same thing a mathematic person will see it in the form of a graph as this is a vertex and this is another vertex, and you connect it with an edge. Okay, so this is the mathematical point of view. So this is nothing but a graph. Okay, a directed graph, you can see. Okay, this is a graph. And the same thing can be put in the form of a matrix like this. Okay, this reaction can be put in the form of a matrix like this. That is, see, when a molecule A, that is, you say a substrate is converted to the product by the action of this enzyme, means some amount of substrate should have been lost to form this product. So I'll write this column. So you say this is a row and this is a column. So this column stands for a reaction. Okay, and there are 2 here reactants here. One is A and the other one is B. Okay, here I have got an A here at this place, and here there is a B. So what happens here is this is -1 because this is consumed. The A is consumed to produce B. So here B is produced. So I have a +1 here. This is for a single reaction. So even for a glycolysis, so glycolysis will have so all these reactions, glucose to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6. So everything you can put this in the form of a matrix. Okay, even you can include this ATP, ADP also when you form this matrix. Okay, so this is single reaction. Hope it's clear. So molecule A is consumed to produce molecule B. Okay, A is consumed to produce B. That is, for A you have -1, and for B you have +1, and this is each row stands for the reactants, and this column is for the chemical reaction. So this is what this matrix, this is in the form of a matrix. You call this to be the stoichiometric matrix. Now, see, when you have a when there is a matrix, you can find many properties. Using the properties of matrix, we can find many biological things. So what you hear is mainly the stoichiometric models. They are linear algebra based, and they depend on the law of conservation of mass. So you know what is law of conservation of mass? So for example, see wood, when it is burnt in oxygen, this will give you ashes plus carbon dioxide plus water. Okay, so mass of wood and oxygen will be converted to mass of ashes, carbon dioxide and water. That is, I say that input plus generation minus output plus consumption will be some accumulation. Is it not? So whatever a process happens, your input plus generation minus output plus consumption is equal to accumulation you have here. Now for a steady state process. So what do you mean by a steady state? So when I say a steady state, it is independent of time. Okay, it is independent of time. In all the previous two models, what you have seen, it is dependent on time. You took it as dx by dt, that is in a particular time. Okay, in a time interval you have taken. Now this model, this stoichiometric model, it talks about steady state. Whereas when you go for kinetic model, it is dependent on time. Okay, so this steady state, that is your stoichiometric model talks about the steady state. So in the steady state, your accumulation will be equal to 0. So this value will become 0. So what happens is my input plus generation will be equal to output plus consumption. This is the base for your stoichiometric model. And this stoichiometric modeling, this was a recently developed one, only in the early of 2000, or in the late of 96, 98, only during those period, this stoichiometric model was studied. Okay, and this, if you put systems biology, the name given to these type of problems is, it is systems biology. Okay, if you Google, you can find a lot of papers in the systems biology also. So this is the assumption made in this model. That is your law of conservation mass. That is input plus generation is equal to output plus consumption. Now I'm just taking some example, a very small network I'm taking here. So here my A, B, C, D, E. So these are the reactants I have. A, B, C, D, E, each stands for one row. Okay, and now, and how many reactions are there? I've named the reactions as R1, R2, R3, R4, R5, R6 and R7. So there are 7 reactions. So I have got 7 columns here. Okay, I have 7 columns here. Okay, there are, you got 5 reactants and there are 7 reactions. Now my first reaction is, you take it as a cell. Okay, so this is an input here. Now what happens here is, in this reaction R2, my A is consumed to give B and C. Okay, so it is not a reversible reaction. This is not a reversible one. So A is consumed to give B and C. Now my reaction R3 is, it is reversible. It is a reversible one. B is consumed to give C and C is consumed to give B. Okay, and here, so that when I'm writing the equations, actually, either you can take it as 2 reactions, because it's a small network. Then there is only one reversible reaction I've considered. It doesn't matter much whether you take it as a single reaction also. Okay, now my reaction R4 is, C is consumed to give D. Okay, my R5 is again an input here, and this is my R6 is, A and E together gives D. Okay, A and E react together to give this product D. And my R7 is, this D goes out of the cell. So this is the reaction I've taken. So this network, okay, this, when a biology person, when they say this, they can understand this easily. Now this is transformed into a mathematical form here. So this network is transformed in the mathematical form. So if a person understands this concept, when I'm given this matrix and if you're asked to form a network, they can do it easily. From this matrix, this network can be formed, and the vice versa also. From this network, you can write it in the form of a matrix. So my first one is A. So A comes here. So that is plus 1. And there is no other B, C, D, E. Everything is 0. And when you take this reaction R2, my A is consumed. Whenever there is a consumption, you have a minus sign. Okay, so I have a minus 1 here. And from A, I'm getting the two products B and C. These two are produced. So I have plus 1 and plus 1 here. Okay, here it is a plus 1 and here it is plus 1. And this reaction has nothing to do with D and E. So the values are 0. Okay, and my third one is I'm considering it here as only one way, or you can have one more column and you can take the reversible part also. So here I've taken my B is consumed to give C. So whenever there is a consumption, you have a minus 1, and whenever there is a product, it is plus 1 here. So my B is consumed, so there is minus 1 here, and C is produced, you have a plus 1 here. Okay, and all other, my A, D, E, they have nothing to do with this reaction, so the values are 0 here. Okay, and similarly when my C and D, C is consumed to give D, so I have this reaction, and then my R5 is again, you have an input of E here, so it is plus 1, okay, and then my R6 is my A and E are consumed, so minus 1 for A and minus 1 for E, and they both consumed and they give the product D. Okay, you have a D here. Now this D is getting excreted out of the cell, so I have a minus 1 here. So this is a matrix with respect to this network. Okay, so this is a matrix you get with respect to this network. Now the mathematical properties of this matrix, they will give you some feasible values of this biological network. You can find some significance you can have for this biological network. Now what you do with this is, I'm considering this. So mathematics people, if you have PG students here, they'll be knowing that they would have studied about linear transformation and linear algebra. So this, yes, I'm taking it as a linear transformation from the flux vectors. So what do you mean by fluxes? See this V you call it as flux, Flux, the flux vector. Now what do you mean by this flux vector is the rate of change of A to produce the, suppose if I take the rate of change of A to produce B and C. So that is my flux. Okay, flux is the rate of change of the substrate to give the product. So that is my flux here. Now I can have this matrix. So S is my stoichiometric matrix. The name of this matrix, we call it as the stoichiometric matrix because you have the stoichiometry of the reactions here. So this is my stoichiometric matrix. Now this equation. So maths people will be knowing it. That is, it's a linear transformation. So this is nothing but the null space. Your S dot V is equal to 0. So V1, V2, V3, V4, V5, V6 and V7. So we had 7 reactions. So V1 is the flux related to R1, V2 is the flux related to R2, like that. My V7 is the flux related to R7. So I have this equation. So S dot V equal to 0. So I can solve this system of homogeneous equations. Okay. So this system of homogeneous equations, so these are the equations. So even whether you're maths or biology students, you will be knowing how to multiply a matrix. So 1 into V1 minus 1 into V2 and remaining a 0. I get these equations. So I can solve these equations. Okay, so I have two linearly independent solutions for this. So that is, if I substitute, this is my V1, V2, V3, V4, V5, V6 and V7. If I substitute these values in this equation, my right hand side should be equal to 0. Okay, my right hand side should be equal to 0. That I have to get it here. So these are the two solutions for this, linearly independent solutions for this. Now this is corresponding to V1, V2 like this here. This is the second solution. Now what does the second column represent? So these are the solutions I've got. So maths people will be knowing about this basis and other things. Biology people don't bother about it. You just say that they are nothing but the rate of change of A to get B. That is your flux. Okay. Now the second one, the second column, it's this 1112002. So 1112002 is nothing but, so this is my V1. That is for my reaction R1. Okay. V1, that is for here, and this is my V2, that is also 1, and this is my V3, and then here it is V4, and then last value is V7. So from A, I get D. So here my overall reaction is A gives two, there is one molecule of A gives two molecules of D. This is my overall reaction here. So this column matrix, okay, it represents the pathway what you had here. From this here, this is one of the pathways. Is it not interesting? So this column matrix, that is the solution of that S dot B equal to 0. This gives you this path. That is from this substrate A, how do you get the product? And also it gives the that 1 mole of A gives 2 moles of B. That also you get it from this. Now again, when you consider the second column, why you're not taking the second column as it is mathematically this solution is right, but biochemically this is not a feasible one, because this -1 -1 -2, okay, we are not considering any reversible reactions here. So these values, this -1 -1, that is V2. So V2, when I say this is with this one, so I cannot have a negative sign here. So -1 -1 -2 2 2 0. This is mathematically it is right, but biochemically it is not a feasible one. So again you have in linear algebra, that is the linear combination of two solutions is again a solution. So the linear combination of two solutions is again a solution. So add the first and second column. So while adding these two columns, I'll get like this. So when you add the first and second column, I'll have 2 0 0 0 2 2 0. So this is for your V1. My V2 is 0, V3 is 0, V4 is 0, and this is my V5, and this is V6, and this is V7. So V1, and then V5, V6, and V7. So this first column represents the pathway. A and E combined together, that is, gives you D. The substrates A and E react together to give the product D. So this is my second one. So when you see this network, the second one is there are two pathways in this. Okay, if you get any other pathway here, they are all nothing but redundant. They are of no use. Okay, they are all redundant pathways. It's of no use here. Okay, they are all redundant. Only from these two pathways, you have from the substrate, you get a product. Even if you take this as your B gives C and C gives B, if you consider two equations, you have got one more column here as a solution, whereas it gives you B gives C or C gives B. But this is not of any use. It's not you're not going to get any product from that. Okay, so this two pathways are clearly represented. So these two pathways are clearly represented here. The matrix, what you got, is represented in the form of two pathways here. So the first one pathway, in this the first basis vector, which gives one molecule of A gives two molecules of B, whereas in the second pathway, second basis, that is one molecule of A plus one molecule of E gives only one molecule of B. So from this, so which one you will be beneficial for you in case of energy generation? I prefer my first pathway. Okay, so this is just a basic thing I've done, but using this, many more concepts are there. That is elementary mode analysis. You can go for enzyme subsets. All those will be of more biological significance. If any of you are interested in working in this, you can refer to you can Google it, and also you can find out, or if you could contact me also, I can help you in this regard. If you've got PG projects, it's a very good topic. Now, what are the applications of this metabolic models? Why do you apply this metabolic models here? It's again to find the robustness of a network. So how large is your network? See, I've shown you the third figure in the network. It's a very, very, very complicated one. So much of pathways put together. Okay, to find the robustness of this network and to analyze the time-dependent behavior. When you say time-dependent behavior of a network, that goes for the kinetic models. Okay, your kinetic models are time-dependent. That is using a Michael-Mertens constant. And then this is to test a set of enzymes for production of a desired product. Even here you can say that here this one molecule of A here gives 2 moles of B, whereas here this 2 combined together gives only 1. So I can use to test a set of enzymes for production of a desired product, and then to identify the redundant pathways. So in the basis vector, whenever you start from a substrate and you get a final product, only those pathways will be useful. The other pathways will be redundant. Okay, and then this systems biology, it is much used for the drug discovery. Okay, drug effects by analysis of enzyme deficiency. What you will be doing it is there will be some enzyme acting on this, enzyme acting on this. If I don't want this product to be formed, so I can inhibit this enzyme. Mathematically, you can do this inhibition of this enzyme. That's a bioinformatics work. Inhibition of the enzymes is a bioinformatics work. Docking will be useful for that. It's an in silico method. And even a wet lab also you can do using that. And to identify what are the potential drug targets, that is, what are the enzymes I can inhibit, will be inhibited to find the drugs. And then to what happens when I insert an enzyme, and what happens when I delete an enzyme. All those studies you can do using this metabolic model. And again, it helps biologists for wet lab studies also. This mathematical model will help the biologists during their wet lab studies. And then, and mostly the stoichiometric model, it's very much helpful for in kinetic modeling when the experimental data are not available. So suppose if I want to go for kinetic model, and if 1 or 2 particular Michael-Mentens constant, those values are not available, in that cases, I can use this stoichiometric model. These are the applications of this metabolic models. Okay, I think I have another PPT also to go, but it's already 1 hour. So I think it's enough. So you have seen the 3 types of model here. There is predator-prey model and epidemic models, and then your metabolic network models. So just we have not solved anything or gone much deeper into this. Just I've given a glimpse of how to how to construct a mathematical model and what are mathematical models. And it is not, mathematical modeling is not at all complex. Once you get into that, it will be very easy. And if and it's good that both mathematics and biology students are here. If biology people and mathematics people, they combine together, they can work a lot in these type of models. So thank you.